Hi, this is Sal,
And Jay.
and today we are going to be explaining the process of cellular respiration. The purpose of cellular respiration is essential for life. It turns the chemical energy stored in food, especially glucose, into ATP. ATP is the energy currency that is required to function or even run the cell. Without ATP, muscles can't contract, neurons can't fire, and life basically cannot function if ATP is non existent. All living organisms perform some version of cellular respiration. Most plants, animals, fungi, protists, and many bacteria use aerobic respiration, which requires oxygen. Some bacteria perform anaerobic respiration. However, even humans are able to use anaerobic respiration when oxygen is scarce, like in overworked muscles. The cells switch to fermentation as a temporary backup. There are two main ways ATP is made during respiration. First is the substrate level phosphorylation. This is where an enzyme transfers a phosphate group to ADP. It happens in glycolysis in the Krebs cycle, but it only produces a little ATP. Second is the oxidative phosphorylation, and this is when the electron transport chain and ATP synthase work together to create most of the cell's ATP. It requires oxygen and a proton gradient across the inner mitochondrial membrane. Cellular respiration relies on redox reactions, which are reactions where electrons are moved. One molecule gets oxidized, which means it loses electrons, while another gets reduced, which means it gains electrons. During respiration, glucose is oxidized and oxygen is reduced. Lastly, you wonder how we are able to create ATP so quickly for even our daily expenditures. It is all thanks to NADH and FADH. To move electrons efficiently, the cell uses electron carriers NAD plus and FAD. When they pick up electrons and protons, they become NADH and FAD2 which are reduced forms because they gained an electron. They carry these high energy electrons to the electron transport chain, and there they release the electrons, become oxidized again, and help the ATP production. However,
This complex and well articulated system can fail with a couple drops of a drug called cyanide. Cyanide is an extremely lethal toxin found in some plants, industrial chemicals, and smoke from fires. It's dangerous because it targets the mitochondria, the powerhouses of our cells. You might have heard of cyanide from detective movies and books. That is because cyanide is fast acting, and it's very lethal, as I said earlier. During oxidative phosphorylation, electrons move through the protein complexes in the electron transport chain, where oxygen waits as the final electron acceptor. Cyanide will actually bind to one of these complexes, which is also called the competitive binding, and the electron won't be able to pass through the complexes. This will result in the failure of ATP synthesis because the proton gradient would fail. Without ATP, every cell of your body would begin shutting down, causing that quick kill.
Hi everyone. So as in the first video, we explained that cellular respiration was the process of glucose turning into ATP. So today I'll be explaining glycolysis, which is the first step to break down glucose. Glycolysis is an anaerobic process, which means that it requires no oxygen and it happens in the cytoplasm of the cell. So we're just going to draw cytoplasm. First, a 6 carbon glucose molecule enters the cell, and the glucose is phosphorylated, which means that a phosphate group is added to the molecule, trapping the glucose inside the cell and preparing it for breakdown. Lastly, glucose is split into two pyruvates, and here is the equation for this reaction. C6H12O6 equals 2 pyruvates, 2 ATPs, and 2 NADHs. So from one glucose molecule, the cell is going to gain 2 pyruvates, 2 NADH molecules, and 2 ATPs, and that is the output of this reaction. Some of you may have been wondering how glycolysis can be affected by feedback inhibition. And here's how. Feedback inhibition is the process where the end product of a pathway like ATP inhibits an enzyme earlier in the same pathway, which prevents the cell from wasting energy by making more ATP than it needs. In glycolysis, dysregulation mainly happens through three key enzymes, and here they are. The first one is hexokinase, which hexokinase catalyzes the first step. It adds a phosphate to glucose, and it is inhibited by its product, which is a glucose 6 phosphate. This prevents too much glucose from being trapped in the cell when energy levels are high. The second enzyme is phosphofructokinase 1. This is the main control point of glycolysis. It is inhibited by ATP and citrate, which signals high energy, and it is activated by AMP, which signals low energy. When ATP levels are high, PFK1 slows glycolysis to save resources. The third one is pyruvate kinase. Pyruvate kinase catalyzes the last step, forming pyruvate and ATP. It is inhibited by ATP and alanine. It is activated by fructose 1 6 bisophosphate, which is a go signal that glycolysis is active. So when ATP levels rise, glycolysis slows down. When ATP drops and AMP rises, glycolysis speeds up to make more ATP. This system keeps the cell's energy balance stable like a thermostat controlling heat. Without oxygen, NADH molecules need to be recycled back into NAD+. So glycolysis can keep going. And this is where the next video comes in, which is about the two types of fermentation. Here, I will be explaining the two types of fermentation. These two types of fermentation are aerobic, and they happen in the cytoplasm along with glycolysis. The first type of fermentation is lactic acid fermentation. In this process, pyruvate is converted into lactic acid or lactate. This happens in muscle cells and also in bacteria to make yogurt and cheese. And the products of this process is two lactate molecules. The second type of fermentation is alcohol fermentation. In this process, pyruvate is converted into ethanol and carbon dioxide. This happens mainly in yeast, and the products of this process are 2-ethanol plus CO2.
Hi everyone. So today I'll be talking about what happens after glycolysis when oxygen is present. So when oxygen is present, it would make it aerobic. So after glycolysis, each molecule of glucose, in this case it has carbon atoms, it splits into two pyruvate molecules. So as you can see, there's 3 carbon atoms each. These pyruvates in the cytoplasm must be transported into the mitochondrial matrix to continue the aerobic respiration. So the pyruvate oxidation occurs in the mitochondrial matrix after pyruvate is transported through the mitochondrial membrane. So as it so usually it will enter through a protein channel. And now that it's in the matrix, each pyruvate, which also still has 3 carbon atoms, will lose a carbon and release as CO2. So the remaining 2 carbon acetyls are oxidized, which means it loses electrons. So technically there's 4 carbon atoms left, but those are 2 acetyls because each acetyl has 2 carbon atoms. So the acetyl will attach to the coenzyme A and form acetyl-CoA. Those electrons are picked back up by the NAD plus and turns into NADH plus and just H plus. So for TDLR, NAD plus is reduced, which means it gained electrons, and the acetyl is oxidized, which means it lost electrons. So overall, the purpose of this whole step is to really just prep for the Krebs cycle and harvest some electrons with NADH for the electron transport chain. Hi everyone. Now I'll be talking about the Krebs cycle, which is also known as the citric acid cycle, and it is an important part of the aerobic cellular respiration. So, the Krebs cycle is actually also known as the citric acid cycle because citric acid is also produced during the cycle, but in our scope of AP biology, we won't be talking about it here. Anyway, the Krebs cycle happens in the matrix of the mitochondria, and as I said, it's aerobic, so it can only occur when oxygen is present. That is because, even though I'll explain this later, I'll mention it. That is because electron carriers here are produced NADH and FADH, and they must deliver their electrons to the electron transport chain, and oxygen is actually the final electron acceptor there. If oxygen isn't available, pyruvate cannot enter the mitochondria and the cell will perform anaerobic fermentation instead. So, anyways, the Krebs cycle begins with two acetyl-CoA entering the mitochondrial matrix. So the two acetyl-CoA has two carbon molecules each. Okay. So anyways, as the cycle continues, the acetyl groups are oxidized, which means they lose electrons. These electrons are picked up by the electron carriers NAD plus and FAD, forming NADH and FADH. These high energy molecules will eventually deliver their electrons to the electron transport chain to fuel oxidative phosphorylation. So each of the Krebs cycle produces 3 NADH, 1 FADH, and 1 ATP made by substrate level phosphorylation, and 2 molecules of carbon dioxide as waste. So every glucose molecule actually runs the cycle twice, which means one glucose molecule will give us 6 NADH, 2 FADH, 2 ATP, and 4 CO2 will be released as waste. So the whole purpose of Krebs cycle is to get rid of all carbon for the electron transport chain and get all the high energy electrons from the acetyl group to store them in the NADH and the FADH. These electron carriers will then move on to the electron transport chain where majority of ATP is produced. Hi everyone. This is our last step of cellular respiration when oxygen is presented. So it would also still make it aerobic. So electron transport chain and chemiosmosis is our final step of cellular respiration. After the Krebs cycle, all the NADH and FADH that were produced carry high energy electrons to the inner membrane of the mitochondria, where the electron transport chain, which also stands for ETC, takes place. This stage is where most of the ATP is made during aerobic respiration. So, as I said, these processes occur across the inner mitochondrial membrane. The ETC itself is embedded in the membrane, and chemiosmosis happens as protons move through the ATP synthase channel back into the mitochondrial matrix. So, as you can see, the NADH and FADH molecules donate their high energy electrons to the ETC. As electrons pass along the chain, they lose energy, which is used to pump protons from the mitochondrial matrix into the inner membranous space. This creates a high concentration of protons on one side of the membrane, forming a protein gradient. The electrons keep moving through the chain until they reach the final electron acceptor, oxygen. Oxygen combines with the electrons and protons to form water, H2O. This is why oxygen is essential for aerobic respiration. So, the last process that I'm going to explain is chemiosmosis. So, as I said earlier, the protons in the inner membrane space from the NADH and the FADH go back into the mitochondrial matrix through the ATP synthase. This is a special enzyme that acts like a molecular turbine. So, what I mean by that is, as proteins flow through, ATP synthase will actually spin and catalyze the formation of ATP from the ADP and the PI, which is an inorganic phosphate. So, in total, the final products earned from this process by one glucose molecule are approximately 28 molecules of ATP and 3 molecules of water.